Hi guys, welcome to the Live 1 Biology 2019 exam paper. We'll be walking it through this video. So question 1, DNA structure. Label the blank bases A, G, T, or C in the diagram below. So we know that adenine pairs with thymine and guanine pairs with cytosine. So we can fill it out down below. A goes with T, G goes with C, T goes with A, and C goes with G. So question B is, some adults can digest milk, but the majority, 65%, cannot. The ability to digest milk as an adult is caused by a DNA mutation. So what is a mutation? A mutation is a permanent change in the DNA sequence. And because of this change in the sequence, a new allele can be produced. Which causes, rather I should say, causes a new allele. So is question B there? Question C. Explain how a mutation can give adults the ability to digest milk. You should include the terms DNA, gene, allele, phenotype, mutation. Okay. So the ability to digest milk and the ability not to digest milk is dependent on the mutation. So if you do have a mutation, it'll give you the ability to digest milk. So in your answer, you should define all these terms here. And yeah, so we can start with saying a mutation is a change in DNA sequence causes a new allele. So is mutation done? We can go down and say now, talk about DNA, and DNA carries the genetic information needed for an organism. So DNA down there. Gene. So a gene is a section of DNA or a specific sequence of DNA that codes for a specific trait or like a phenotype. In this case, it is the ability to digest milk. So the DNA sequence, which has been mutated, causes the gene to code for the ability to digest milk, if that makes sense. So we can say a gene is a section of the DNA that codes for specific trait. In this case, the trait is the ability to digest milk. So that's gene done. Allele. An allele is an alternative form of gene. So we can say an allele is a alternative form of a gene. So that's gene done, allele done. Now phenotype. This is the last part you have to talk about. And this is where you get the excellence from. Relating the question back to phenotype. So as the mutation changes the order of DNA bases, this causes a new allele, and the new allele codes for the specific trait, so which is a phenotype. And the phenotype is like an outward expression, so the ability to digest milk is considered as a phenotype. So we can write as the mutation causes a change in the DNA sequence, the allele for ability to digest milk is formed. And this allele codes for the phenotype. So there we have it. This question is done here. So first we just explained what a mutation is, then DNA, then we talked about the gene and the allele, and what the gene codes for the specific trait. And lastly, the last part of the question is describing the allele and how, in this case, it traits for the specific phenotype of digesting milk. And that will be an excellent question. So question 2 is on cystic fibrosis, and it is an inherited condition. It can be traced through a family chart shown below. And so unaffected allele is dominant. So unaffected allele, it's dominant, and affected is little t. So as you can see that the two unaffected parents produced 4 offspring, and 2 of them are affected and 2 of them are unaffected. So this means that the parent and the male and female parents are carrying heterozygous alleles, as 2 are affected and 2 are non-affected. If one was carrying a homozygous dominant allele, then all would be affected, or all, in this case, would be non-affected. So it asks us to complete a Punnett squared for the cross between individual 1 and individual 2. So we can just put down T, 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 as both parents carry heterozygous alleles. So doing this will give us TT, T, lowercase t, and 2 lowercases t. So we can just do male 1, female 1. And that's the Punnett square done. Work out the genotypes of the following 3 individuals. So it's wanting us to go back up to the chart and identify which individual has the genotypes. So for individual 1, we can see that it's male 1, so it'll carry a TT allele, as we know that both parents carry the heterozygous allele. So we can just write TT. Individual 2 is the female, so this also carries the heterozygous allele. So we write TT. And these just come here. So we know that both of them are, individual 1 and 2 are heterozygous. Individual 5. So individual 5 is affected. And we know that lowercase t represents affected. So that means that to be fully affected, he must be a lowercase t and lowercase t, homozygous recessive, to carry the affected allele. So T, T. So question 2C, explain any difference in the expected phenotypes of the offspring ratio to the actual phenotypes of the offspring ratio with individual 1 and individual 2. Phenotype ratio. So what they're running in this question is comparing the ratio we got from the Punnett square to the ratio they gave us, which they actually produced in the pedigree chart. So the expected phenotype ratio was 3:1, according to our Punnett square. And actual phenotype ratio, which we obtained, was 2-2. So there's 2 unaffected and 2 affected. So we write 2-2. And this 3-1 comes from the Punnett square here, as 3 of the individuals produced from individual 1 and 2 carry the homozygous dominant allele T, and 1 offspring carries homozygous recessive lowercase tt. That means the ratio is 3-1. So in your answer, you should refer to the expected and active. So what they're wanting is just explaining why this occurred and what are the reasons behind it. So if the actual phenotype has a greater number than the expected phenotype, this means that the parents, male 1 and female 1, carry more of the condition than expected. So we can write parents F1 and F2, they carry more of the condition than expected. So to F1 and F2, I'm referring to individual 1 and individual 2. They're carrying more of the cytistic fibrosis than expected, because what we expected was only 1 of the individuals having the condition, but in reality, 2 of them were affected and 2 of them weren't affected. So we can write that, and then we can write, so this occurs due to the fact each offspring or individual is outcome of a random event. Event, I'm meaning like fertilization. So when the zygote is being produced, it's random, completely random. And this can vary the chances of the parents passing the condition off to the offspring. And we can also write that each offspring is unaffected by the previous outcome. What this means is that the offspring previous outcome. This is just referring to, so this individual here was completely random, and this one here was completely random as well. So when TT was produced, the production of TF, or T, lowercase t, has nothing to do with TT. It's completely random. And that's how the chances of inheriting a condition increase or decrease. So then we can talk about individual in the 1 and 2. So parents, these two, as they both carry heterozygous allele T, the offsprings have a 50% chance. So each offspring here has a 50% chance of inheriting either allele. So they can either inherit T or TT from each parent. So we're going to say since parent 1 and parent 2 carry our heterozygous trait, each offspring has a 50% chance of obtaining either allele as it is t lowercase t. So this is question 3, sexual reproduction and survival. So there's a disease damaging nutrients and tissues within a tree. This means some trees survive and others starve to death. Describe the genetic variation in trees. So the genetic variation is pretty much just the differences in the DNA and genes of each tree. So you can write genetic variation is the differences or variety in DNA. Question 2b. Explain how the sexual reproduction of trees causes genetic variation and diabetic disease. In your essay, you should consider gamete formation, fertilization, how sexual reproduction leads to variation in the population, and the link between genetic variation and the survival of coyote trees as a species. So we can first start by talking about gamete formation. Gamete formation is produced during meiosis, and it is produced by either crossing over and independent assortment or segregation. So we can say gametic formation occurs during meiosis through random assortment. This is the random assortment of chromosomes. Random assortment of chromosomes. Crossing over of chromosomes. So this is when chromosomes cross over and parts of the chromosomes swap with the other chromosomes, so that the gametes formed have no new allele combinations, which produces genetic variation. So crossing over or segregation. Yeah. So these three processes form the gametes. Meiosis, we've talked about. Meiosis produces the gametes. And the gametes which carry half the chromosomes in the parent cell. So this statement here is really important, that the gametes produced carry the half the amount of chromosomes in the body. They don't carry all, they carry half. Next, we can talk about fertilization and how gametes are produced. So here we fertilization. Fertilization is when male, female gametes fuse to form a zygote. Just a quick note that each the female and male gametes have each contain like their unique DNA. So each gamete has their unique set of DNA sequences and carry unique genetic information. because of this, the zygote which is produced is also unique. And so we can say from male both unique in DNA, therefore producing a zygote genetically unique. So that's fertilization done, meiosis done, and gametic formation done. Now we can talk about sexual reproduction. So sexual reproduction is pretty much done to produce new combinations of allele, and this producing new combinations of allele allows the population to survive. And this is what gives the genetic diversity to individuals. If no new alleles were made, then and we had all the same alleles, it'll be really hard to survive against things such as diseases and just things that affect the human health or anything in general. So alleles help us fight towards that and plays a big role in natural selection as well. So we can say that sexual reproduction carries the role to produce new allele combinations. And as we said, these combinations are made in the gametes during meiosis by crossing over or independent assortment. Hence, this therefore, where do we go? Therefore, increasing genetic variation amongst individuals. So that's pretty much the role of sexual reproduction. And because of this, it leads to variation in the population because of the new allele combinations. Now we can talk about the link between genetic variation and the survival of the trees as a species. So we can say that genetic variation is advantageous to individuals, or in this case I think it's species, species to survive. Species that survive pass on alleles to offspring. Generous offspring. So, lastly, we can say that species over time, the genetic diversity will increase amongst population allowing, in this case, the trees to survive. If it was like an animal or a mammal, this will allow the mammal to keep reproducing, and create offspring with the favorable allele, and that allele allows that offspring to survive, and pass those alleles on and on. So yeah, that's pretty much that's all. Thank you for watching, and that's the end of this video. Thank you.